Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Michael Betts. He's a professor of microbiology at the Penn Institute for Immunology, Penn meaning Pennsylvania. He normally studies T lymphocyte function and the role that these cells have in controlling and eliminating viral pathogens and, being a part of the immune system. So, Michael, thanks for coming.
Thank you for the invitation.
Yeah. Today, instead of going into just purely your research, I wanted you to be a part of the virus book I'm putting together. So I was going to ask you a series of questions around that. And again, if you can't answer one, it's okay. No problem. So,
okay.
Before we do that, can you just in your own words, give a little bit of background on your work and just the current research that you're working on right now?
Sure. So my interest is in understanding how humans combat pathogens with the immune responses, and particularly adaptive immune responses, which would be, in this case, T cells and B cells, but mostly we work on T cells. And we've historically focused a lot on studying immunology in the context of HIV infection. More recently, we've, in addition now, are studying, of course, SARS-CoV-2 infection, and also working in areas of really basic human immunology, trying to understand how T cells move around in the body, what happens to them when they go into different places, which then all pertains to how those cells will function in response to an invading viral pathogen or a pathogenic challenge, which takes place really at potentially many different areas of the body. So trying to understand how these cells function at those different sites.
So in regards to these questions, the first one I had is that, do you know of any organism that doesn't have viruses? Do you think that they accompany all types of life?
I am certainly unaware of an organism that does not have some type of viral pathogen or related type of I wouldn't call them an organism, a related thing. So no, I am unaware of an organism that is not subject to viral infection.
Are there any organisms that do have viruses where it amazes you that they do have them, or that the viral action causes the organism to do just really unusual things that sticks out in your mind?
Well, I guess the only place where, going back even to the first question, I guess I'm a little bit unaware of the biology of this, and certainly there would be many people who would have expertise. But I guess if you want to sort of be amazed about something, I would assume that these types of bacteria, which we call extremophiles, would also be subject to viral infections, bacteriophage, and I guess the amazing part of this, if that's true, and I assume it would be, is that the viruses, the viral particles, are able to also remain intact in sort of these extreme environments, such as extreme salt environments or extreme heat environments, where you have these different extremophile bacteria. So that would be probably a pretty interesting place to learn about them.
That's true.
Yeah, I mean, multicellular organisms. I think any place where there's a multicellular organism, you're going to have viruses. So none of that would particularly amaze me, just because it's kind of expected.
And I guess there's viruses that predate on other viruses, giant ones, and then I guess they're called like virophages. So it seems to be multi-level.
Certainly, some of them can become quite large. The pox viruses, for example, are quite large. I think there's some that are even larger than the pox viruses. I honestly have to say I don't really quite know from the evolutionary scale of I would certainly expect the super simple viruses to have come first, and the other ones have probably accumulated their size through interactions with hosts and commandeering things that were beneficial to them. Yeah.
Yeah, that leads into a question later on, but we'll ask it now. Which do you think came first, cellular life or viruses, and why?
Well, I think, some type of replicating forms of DNA or RNA. I think there's a fair amount of research in this area. Self-replicating forms of these things, which I guess in some ways is almost like a virus. But my guess would be, again, I'm not an evolutionary biologist, but my expectation would be that these, if you want to classify these replicating forms of genomic material as a viral predecessor, I would guess that comes first. They would be highly subject to their environment and having the right components around their environment to replicate. That would be my guess on this.
And do you think that viruses are alive, and why or why not?
No, I do not, and that's simply because, in my opinion, they're basically machines, and that's, they do not have classical things like organelles and things that cells, even the primitive cells, would have. They do have membranes, but they take those from the cell that they're budding from, so the types that have membranes. They're never autonomous. They always require something from the host cell. So no, I would not classify them as being alive whatsoever. They have components. They have components necessary for life, like DNA or RNA, but themselves, no.
Well, do you think they could be contingently alive only when inside of a cell, and when they're in virient stage, not alive? Like a seed, we could say, is dormant. It leads to a living thing in the right soil and right conditions. But if you think of a tree that has seeds and that's how it spreads, is the seed alive? And the tree is alive.
Yeah, no, I don't. I think really, the virus is. No, I don't think I would not consider the virus alive even when it's infecting a cell. I think it's a combination of proteins and genomic material that are interdependent, and therefore that they would have to be interdependent for the assembly process to take place inside of the cell and so on. And each of these components has somehow evolved to interact with each other and form these complexes. But no, I don't consider them alive even in that context. They're kind of. A seed is alive. That's the thing. You can take it out, and there are cells in there that will replicate on their own and form a plant. The virus, if it's just sitting on the table, is not going to do that without having. So in that sense, it's a seed and it's not a seed. But I would not consider it alive because it does not have that ability without any assistance whatsoever to replicate.
Well, if you look at the shape and action of viruses, they have many different entry mechanisms into cells and many different shapes. Some are rods or spiked balls, or the T4 bacteriophage is like a moonlander. How do you think that variation occurs, and how could they be so diverse?
That's a fantastic question, and it's. But of course, the answer is nobody knows how that would have arisen. If you think about the shape of the virus itself is essentially meant to protect the genomic material inside, and that essentially you need a self-assembling complex of proteins, and those proteins can assemble in many different ways, and there are many different structures that could arrive at an enclosed space. There certainly are. There's families of viruses that have sort of shared relationships. The way that they work, you have the icosahedrals, you have the moonlanders, as you say, and they're all essentially designed to protect and, as necessary, deliver that genomic material to the cell. So, like the bacteriophages, there's a really cool way that they do this. Essentially injecting the genomic material into the bacteria, And that's all essentially kind of like spring-loaded mechanisms of these proteins that once they get the proper trigger, it just sort of things just snap like a mouse trap almost. You just have this sort of consequential downstream effects that shoots the genomic material into the bacteria, and even the eukaryotic viruses have almost the same conceptual methods. HIV, for example, you have this the envelope and interacting with the CD4 and the CCR5 co-receptor, and that induces this protein conformational change that essentially shoots the genomic material into the cell. So it kind of all, despite the fact that some of them look a little different, they're all functionally related in terms of strategy as to what they want to do.
Yeah, is there any particular entry mechanism for a virus that, I don't know, again, surprises you or you think is really cool? Is there any particular one that you know of?
Oh, I mean, honestly, Richard, every single one of them is fascinating in and of itself. Just how on earth could this particular set of circumstances be built upon to do what it does? HIV, the one I would certainly know the most about it is fascinating in the level of complexity that has evolved on the virus to not only allow entry of that genomic material into the cell, but in the meantime to protect that virus from the consistent pressure of the host immune response, antibodies. You have this conformational changes that take place in the envelope like a protein that there's only certain formations that are ever seen during the entry process, whereas otherwise the immune response rarely ever would see these things. And it requires multiple proteins being involved, and every single one of them is fascinating in of itself, to be honest. Yeah.
Well, HIV is interesting. I mean, so far as I know, it endogenizes into our DNA, right?
Yes, as a retrovirus, yes, it does, and there's other types that do that as well. In the retrovirus family, there are other viruses that form stable episomal circles. There's some, like flu that doesn't do that. It just sits there and has all its different strands and makes the viral proteins from multiple strands. So, different strategies. It sort of depends on, and this sort of addresses some of the other questions you have in the list. That some viruses have almost a hit and run type of strategy. Get in, make some more virus, and get out before the host responds. And others want to set up shop and be there for a long time, like the retroviruses, which integrate, or the herpes viruses, which form these sort of stable genomic circles, or maintain like true latency, like Epstein-Barr virus, and just sort of sit there in the cell.
What would govern that? Like, why would HIV endogenize and other ones just want to, multiply and lyse the cells, and then the latent ones or the lysogenic ones are the strangest because they'll be latent days, months, weeks, years, sometimes lifetimes, and then when the host encounters stress or certain other problems, now all of a sudden they turn, let's say, lytic. Why do you think that would happen? How could the virus be monitoring, or is it monitoring the host's immune state?
Yeah, I mean, there's a whole lot of different answers to that, because every virus is kind of wanting to do a different thing. I mean, ultimately, the goal of the virus simply is to propagate, and whether it does so at the cost of the host or not, kind of depends on the virus in some senses. Some viruses kill you because they're replicating so quickly, or they're so pathogenic for other reasons, and others don't kill you or make you sick at all, or seemingly don't make you sick at all, yet they're all still propagating. If you look at some of the most successful viruses that are truly can infect you or pathogenic, so we actually have endogenous viruses in our genomes. That if you work really hard, you can force them to make particles, but we've just lived with them for millions of years, and they're just in our genome all the time. Whether they were previously, pathogenic, nobody really knows. But something like Epstein-Barr virus, something like 90% of people are infected with that, and we pretty much all live with it. the virus certainly transmits from person to person, but we just, and I don't know that people get any benefit from being infected with Epstein-Barr virus, which addresses another question you have in there. But there can be some benefits to being infected with things.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yeah, what are some? Like, I know, I guess viral DNA is responsible for a mother to be able to create a placenta that separates the fetus from the mother immunologically.
Yeah, so probably the best example of why being infected with things and surviving, of course, obviously you always want to survive, but there's absolutely a very clear example of where having a lifetime of exposure and success in combating whatever the pathogen is, be it viral, fungal, bacterial, whatever. Would be the work that people have done using laboratory mice that are either kept completely pathogen free or allowed to become infected sort of naturally with things. And so this is work by Dave Massapust and out of Minnesota, and there's another group in Germany that also worked in this area. And basically, what happens is the mice that have a lifelong history of exposure to pathogens, and certainly including viruses, whenever they get challenged with something new, they have a much better chance of surviving it than does an animal that's never seen any pathogens at all and suddenly gets hit with something pathogenic. And it's essentially, in some senses, it's thought that the immune response in an organism is kind of always on a knife's edge. And if you, in terms of pathogenesis or protection, because the immune response will both get rid of the pathogens, but it does so in many ways in kind of a blitzing style that can cause collateral damage. And so when you have a lifetime of exposure to viruses that are apathogenic or mildly pathogenic that have evolved with us, or with the organism in question, to not kill it, it trains the immune system in these organisms to not overreact to things. You essentially sort of raise, you could call it tolerance or whatever. You sort of raise your threshold of danger in a sense. This is not the danger idea is an old concept, certainly not my concept. But yeah, I mean, certainly there is a benefit to the host to survive these things, and there's a benefit to the virus to not kill the host.
Well, what about if you're infected with a virus and you're a carrier? It doesn't hurt you for some reason, but your competition, it kills them. So let's say you can have progeny, but other people you come into contact with, it may kill them, and then in that way it may act to arm you with a weapon that allows you to procreate, but not others.
Sure, absolutely, and obviously that would be a passive issue, but sure, and there are certainly examples of where that's been used historically to certain people's benefits and other people's detriments, with biological warfare. But really, if you look over time, millions of years, and you can see, there's evidence that viruses and in this case humans co-evolve with each other, and while viruses might have a very high ability to adapt and evolve, the regions of the human genome that are responding to those viruses are also the most highly adaptive and evolving genes in our chromosomes.
Say more about that. What do you mean specifically?
Well, so there's the best example would be this family of proteins called apobex, and I can't remember what apobex stands for, but there's a whole family of these genes. For example, and a great person who works on this is Paul Benash out of Rockefeller. And so this is a family of proteins that can interact directly with viral genomes in a non-specific way. So they would interact because the viral genome is not like the human genome. It has features of that the immune response can recognize as foreign. And so as the viruses evolve to evade those, these, for example, APOBEC genes can evolve over time. Of course, it's not going to be as fast as what the virus can do, but within the human population, there'll be enough variation in these genes, the kind of in the scenario that you set up, that one person might be protected and the person next to them may not be. This is kind of like the example of where that variation in these host protection genes would allow some people to be protected and others. And then in terms of hundreds of thousands of years, if you have continual pressure from this virus, you would select in the human population for people who have those protective genes. And at the same time, the virus is slowly evolving or adapting to the changes that the human genome would be making.
So in terms of, let's say, HIV, when it becomes part of our DNA, where does it become part of the DNA? And has there ever been an example of it, deendogenizing and packaging its own virions, or does it just continually do that, for instance, with HIV, even after it's endogenized?
Oh, well, that's so as far as we know, the virus, HIV, has to integrate into the genome in order for it to make progeny. That has to happen. So that's what you said is exactly what happens. And we have a paper just about to be published from our own studies of integration, where the virus integrates into the genome. Lots of other people have worked on this for a number of years as well. And basically the answer to your question is, in the case of HIV, the virus primarily likes to enter, to integrate into regions of DNA that are open, so open chromatin. So first, it can't pioneer very effectively and go into closed chromatin. So it looks for open genes, and it seems to prefer open genes that are being, in many cases, actively transcribed. In other words, if it integrates into a gene that's open but not doing anything, there will be no transcription of that gene, and therefore no transcription of the virus. So no progeny. If it goes into a gene that's open and being actively used, then that cell will be producing virus. And actually, that might end up killing the cell, because now it's making a lot of virus, and the virus will eventually kill the cell, or the immune response will recognize that cell and kill the cell. Yeah, integrating into either a non-expressed but open gene or a low-expressed gene might then favor kind of this slow profile that you have with the retroviruses, where some cells get integrated but don't make virus, and they can sit there for years because those cells are very stable, and they can even undergo a process called homeostatic proliferation, where you have the integrated virus in the cell. The cell is resting, but over time it divides very slowly just to keep itself stable over time for the lifetime of the host. And so when it does that, it will actually carry the virus into the daughter cells. And that's been shown by John Coffin's group at Tufts and Frederick very effectively, that this is a way that the virus can perpetuate itself within the host.
So even though, for instance, HIV is endogenized, it still retains its ability to create virions and do all kinds of things. And in a sense, it creates its own, it maintains its identity as a virus. It doesn't become part of us, and then
absolutely,
amazing.
Yeah, absolutely. And we actually have other endogenous retroviruses that are distantly related to HIV in our genomes that are with us for millions of years. These are called HERVs, human endogenous retroviruses. At some point, they were probably pathogenic and transmitted, but they do not seem to be so anymore. And they have deletions in parts of their genome, so they're not full viruses anymore, but they can clearly be recognized as being a viral origin. And it's actually thought, interestingly, that these HERVs and other sort of related things, line elements and things, actually are part of what has allowed us to evolve to be where we are. So in these kinds of situations, you would envision that these retroviruses were carrying a gene that they needed for themselves, but also turned out to be beneficial to the host. And so we were better off having them and being infected with them, and so they became sort of a stable part of our genome. And maybe that's sort of partially how humans have evolved to the current state.
Has there ever been an example of a virus deendogenizing, either during active infection or later on? Like HIV.
Well, I mean, you're kind of, it's almost kind of like a cure question, right? So in other words, can you make that virus leave the genome permanently? There's a lot of people who would love to know the answer to that.
Well, you might be able to CRISPR-Cas9 it out.
Yes, of course, and there are people working on that as a cure strategy. That the problem with that strategy is how do you deliver the CRISPR-Cas9 to every cell in your body that could be potentially infected? But absolutely, it is a viable hypothesis to try to do that. The delivery is the problem. I have to say, no, I don't know of an example, a definitive example where you can show that the virus actually, an integrated virus, truly leaves a cell without an intervention like a CRISPR-Cas9 type of situation. I don't know of any natural way that would happen.
Well, I just thought of something kind of, right now it's science fiction, but what if you identified a HERV and you knew which genes to add to it to make it active again, and you used a therapy, a CRISPR-Cas9 therapy to, or some kind of viral vector to endogenize the missing pieces for a given virus that's already been endogenized into you to make it active again.
Yeah.
I don't know. That'd be crazy if someone did that. It would probably be really bad news, but
it might be dangerous. it might not work. I think there's actually a fair number of people that think about these things, and even in the case of the HERVs, there is a line of research looking into HERVs and targeting HERVs as a way to deal with HIV. It hasn't been terribly successful yet, but basically what you can find is that in HIV infected cells, you can sometimes also see HERVs becoming expressed because there might be some shared reasons for them, components of their genome to become reinvigorated, I guess you could say. Wow. And then so there's some thoughts of using the fact that HERV proteins can be expressed in HIV infected cells to target those cells. The reason why that could theoretically work is that those HERV proteins do not have the same mutation rate as what HIV would. So they would be more of a stable target. That's kind of the concepts behind that strategy. It hasn't borne out quite yet. I think people are still working on it, but it still remains a viable strategy.
Has anyone mapped HERVs and looked for analogs, in active viruses out there? I mean, people always say, 8% of our DNA is viral DNA. Well, how do they know unless there's an analog out there? It's just.
Sure. I mean, I am certainly not an expert in that level of this, but absolutely, just in the human genome, these things have been mapped. And then you can just look at the sequence and you can align that to sequences of things that are out in real life still. And absolutely, you can find you can put them into the families of other viruses that are related to and so on. But I can't give you any crystal clear examples here, except to say that for sure people do it, and there are definitely relationships between the HERVs or the line elements or other things of this type, and things that are still so to say in circulation.
You have to help me find a HERV master that I could speak to in the future.
I'll try to find one. Well, Doug Nixon at Cornell Medical Center has done all the HERV work in the context of HIV, and he would really be able to tell you much more detail on this than myself. Yeah.
Okay. Well, continuing on, when people get infected with a given virus, there seems to be a latency period.
Yes.
Days, weeks, months, sometimes years. Why do you think that exists? Do you think it's just the virus needs time to replicate, so there's quote unquote enough cells infected, or? Yeah, exactly. I mean, I don't know of any example where it's years, but I think some of them certainly replicate very slowly. Others replicate very fast, and there's certainly a threshold
after which, if the virus is replicated enough, it will overwhelm the innate host defenses that are present. So every cell kind of has resistance mechanisms, and you're going to need to overcome that in order for the virus to replicate. It has to overcome these innate defenses that it has, and probably a high proportion of the infecting virions in any case don't surpass those thresholds. But then, really it only takes one to make it past the defenses, and then if that cell starts to produce more virus, then you're infected. And so the delay time is related to both the innate responses and surpassing those, plus the critical threshold of having enough virus to cause symptoms. And symptoms, of course, is really only manifested by, that's the manifestation of the immune response against the virus.
Okay. Well, once a virus is inside a cell, if you imagine it as someone sitting in front of a huge control board, the virus does seem to co-opt cellular machinery, various ones to do its bidding. Do you think that they can use cell-to-cell signaling, extracellular vesicles or anything like that to communicate?
Yeah, I saw that question there. So I don't know, if you want to invoke communication, you're kind of asking whether the virus is alive. I don't believe that the virus.
Bacteria do quorum sensing. I wonder if the virus could do some kind of quorum sensing where it says, okay, how many other infected cells are there in my local microenvironment like me?
Yeah, so I agree with you that bacteria do this. Parasites can do things like this. In both cases, those are live organisms. Here in this case, viruses can affect their environment for sure by changing the cell that they're in. For example, some viruses will they co-opt the host cell protein machinery, and therefore the host cell stops making proteins of its own, and it gets co-opted to make nothing but viral proteins or a high proportion of viral proteins. That might cause then that cell to be sensed in its local environment in a different way, because the cell surface proteins may change. Maybe it becomes detached from a sort of a basement membrane or whatnot. But I don't know of sort of, other than the local environment recognizing that something funny was going on with that cell. I don't think a viral infected cell wouldn't be talking to another viral infected cell and saying, hey, let's do something together. That's not going to happen. You can have situations like there's a thing called syncytia, which is basically where one cell merges with another cell, which merges with another cell. So you get this big multinucleated thing that's a cell, but it's the conglomerate of cells, and viruses can cause that to happen. So I guess in that sense, you could imagine sort of a scenario where you're talking about where one infected cell basically meets up with another infected cell, and they merge and do on, but they're not really in a sense communicating. They're just kind of aggregating.
Well, if you did an experiment, I don't know if this has been done, but if you did an experiment where you had some viral infected cells, and then you looked at their profile of EVs that they were putting out, and you saw, let's say, viral DNA in most of them or all of them, maybe then you could say, well, it looks like at least the virus is causing the cellular machinery to put out more of itself. Maybe it could be a method of spread. I mean, I don't know. If not communication.
You mean extracellular vesicles, EVs?
Yeah. What if it changes the payload of them?
Well, certainly that happens. Certainly there are EVs that come off of every cell, probably, whether it's infected or not. And I guess I would say here that I doubt. I don't know for sure, but my strong feeling here would be that EVs are not, unless they contain the full genome of a virus. Maybe that's possible for some RNA viruses. Maybe an RNA virus could transmit that way, but I think it would be hard pressed for an EV to be infectious. I'm not saying it's impossible, but I think it would be challenging.
Well, what if you compare an EV to a virus? They contain genetic material. They can readily enter into cells. They can change gene expression. I mean, in a way, they're like a virus with a different hat on.
Yeah.
Not exact, but they're somewhat similar, maybe.
Right. No, I get where you're coming from. I mean, very few viruses are only a protein shell and the genome. Most viruses that I can think of are much more complex composition than that. So you might carry part of the necessary transcription units with you, or you might bring with you components that allow you to initially evade the first host immune responses, the intracellular responses. Something that gives those viruses a leg up a little bit whenever they get into what is really a very harsh environment once they enter a cell, because there's proteases everywhere. Basically, all kinds of strategies that cells are going to use to get rid of anything that doesn't belong. And I don't know that the EVs would be able to effectively deliver the genomic material in any type of focused way, for sure. It would just be totally random. Like I said, I'm not saying it's impossible, and this is kind of one of these things that if you believe it to be true, you could probably find a situation where it happens, but I don't know if it's a primary mechanism that's used.
Okay. Yeah,
Oh, it's a fantastic question, and I think it's going to be one of these case-by-case basis where, if you talk to 20 scientists, you're probably going to find some that say, yeah, absolutely, it could happen. And then others that'll say, like me, will say, well, maybe it'll happen. Then you'll probably find some that'll say, absolutely not.
Yeah, that's what I'm finding. Yeah.
Yeah.
Well, the reason I ask is that I've seen viruses, obviously use cells as tools. I've seen cells use viruses as tools, like bacteria pulling in pieces of viral DNA and integrating it into their genes, and then expressing spike proteins themselves and poking holes in other bacteria. So viruses are weird. They're like this vast floating cloud library of ability, but yet they also, have, I don't know, I guess I anthropomorphize it, but they have their own agency because they enter into cells and change them and cause them to do all kinds of things. So it's weird. They are a tool, but they use tools as well.
Yeah, I think being an immunologist, one of the most fascinating types of viruses out there for me, actually more fascinating than HIV, are the herpes viruses. And the reason why they're so fascinating is because they've evolved essentially to carry copies or analogs of our own genes that they have then modified or somehow have gotten a modified version of, to allow them to evade immune responses or to modulate immune responses. Some great examples, and it's maybe a little complicated here, but they have analogs of cytokines that our immune system uses basically to sort of marshal responses. But they have analogs of these cytokines that an infected cell will start to express these viral proteins that are just they're a copy of the cytokine that's just a little bit wrong, that then does something different and essentially steers the immune response in the wrong direction. It's really quite fascinating that this has evolved. And there's multiple examples of these types of things. It's not just herpes virus, pox viruses also do this as well. Kind of the bigger the virus is in terms of genome size, the more it's co-opted and included aspects of the immune response as a way to subvert the immune response and perpetuate itself. It's really fascinating.
Is there any way to, and it just sounds silly, but tell how old a virus is or how long it's been in existence by looking at its DNA and capsid and everything? Are there some that we know for sure predated others?
I mean, without having, so I'm not a great person to answer that question. Certainly there, you can look at viral origins. This has been done very well, for example, with HIV. So Beatrice Hahn at University of Pennsylvania has looked at the origins of HIV for years, going back into the monkey variants, the SIVs, and looking to see where those, and how did that come to humans? And in order to do that, she would look for the associated virus in the monkeys, and then compare its genome to the human genome, to the virus that infects humans. And then basically do comparative evolutionary biology on these and really try to understand, okay, what changed in this virus to allow it to infect humans? And so she spent a large part of her career studying this. And you can do this for many other viruses as well. Oftentimes, you want to go back to even fossil samples and try to find things or whatnot. But I'm not an evolutionary biologist. There definitely are strategies you can do this, but you need to look at related viruses and then compare them and look at how things diverged over time. Then you can calculate time. If you know like the mutation rate of a virus, you could then confer backwards if you know related species that differ by a certain percentage, and you can track back in terms of millions of years to find what a common ancestor would be.
So if I get infected by a virus, and let's say somehow I'm the first one, I wander into the jungle and zoonotically something jumps into me.
You're probably going to die, but yeah.
Here comes the question. So if I label myself number one and I go back to my village and I collapse and like cough on somebody and they're number two, they get infected, and then 3 and 4 and 5. By the time it gets to number 20 or number 100, what do you think will happen to that person versus me, number one, and why? Well, how do you think the virus will change?
I mean, in that kind of scenario, I imagine the virus isn't changing much at all. Certainly there is some variation from person to person, but I think in that scenario, that rapid level of transmission, the virus is not going to need to change very much. Evolution of viruses takes years. Between people, like if you think about flu, there's seasonal variations every year. There's shift and there's drift.
I thought like HIV changes unbelievably.
Well, you have a quasi-species in it, yes. So you do have quasi-species within a person, and you have diversity within the HIV across people. And yes, indeed, within one person, you have a tremendous level of diversity. But in order for that diversity in one person to shift an entire population, it still takes a long time. It has to be transmitted from host to host to host. You've got competing host immune responses going against it, and you've got the variation in those host immune responses, which will differ from person to person to person. And so in the case of HIV, you may have a more difficult time to go into one host and an easier time to go into another host, depending on the level of differences between those people at the genetic level. So, in your scenario, my guess would be, no, that virus probably doesn't need to mutate very much. It's probably little benefit for it to kill its host so quickly in the long run. But in the short run, it's propagating. And that's, if you think the virus cares, that's all it cares about, is propagating.
All right, last question. If I get a virus from someone that's like on death's door because of the virus, and versus if I get it from someone that, doesn't feel anything, they're asymptomatic, do you think I'm going to get very sick if I get sick from the really sick person, where there's so many factors that make me that we know correlation?
Of course, it depends on the nature of the virus, but no, I don't think that you can immediately predict. COVID here is the perfect example. We have no idea why some people have asymptomatic infection and others get sick and die. We really just, at this point, we don't know what that association, this is getting into sort of host genomics. And certainly there are some associations with different genetic polymorphisms and disease severity for many different viruses. And there's some emerging information about SARS-CoV-2 as well. But no, I think it's unless, in your scenario, you have the virus has a display of being completely apathogenic or being pathogenic. So therefore, no, you can't tell. If it was always pathogenic, then you're probably going to have pathogenesis. But I think it's difficult to predict. Now that doesn't say it's going to be forever impossible to do so. Certainly there are genetic polymorphisms in the human population that will predispose you to be more susceptible to things. Particularly if you have polymorphisms in various immune response related things, that can be very problematic and therefore be a predictor of pathogenesis.
Yeah, Mike, I just want to thank you for coming, and I appreciate it. How can people find out more about your work in particular?
I have a website, which is betslab.org, and people could read up on my lab and my lab members and even see some of our work, and that would be a great place to start.
Very good. Well, thank you for coming, Mike. I appreciate it.
Yeah, thank you again for the invitation.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.